Hi there, this is going to be the second half of our first lecture. This is Chapter 2, the chemical basis of life. Let's go here. This is our little introductory slide giving you a whole lot of details about what we're going to be doing in here, running through the main concepts of things we're going to approach. But before that, I'm going to run you through this hierarchy of complexity one more good time, just to make sure we understand the concepts. You know that you are an organism made up of many organ systems, digestive system, respiratory system. These organ systems being made up of organs. These organs being made up of tissues, that would be several tissues coming together to make an organ. An organ is a group of two or more tissues working together towards a purpose. A tissue being a group of similar cells working together towards some sort of purpose. Tissues being made up of individual cells, cellular structures, cytology, tissues, histology. These cells being made up of individual organelles, like your mitochondria, nuclei, etc. Organelles being made up of macromolecules, like proteins, like lipids. These macromolecules being made up of smaller molecules bound together. And then these smaller molecules being made up of individual atoms. So, if we're going to talk about how your body works, we're going to talk about atoms at the base of this. So let's talk about chemical basis of life. Here we go. First things first, matter and energy. Everything you can see, touch, or feel is matter. Matter comes in various forms, solids, liquids, and gases. But for our purposes, everything we see, this board is matter, this structure is matter. I am made of matter. This table is matter. The batteries inside of here, they are matter. My shirt is matter. The air I breathe is matter. It is all made up of matter. Ergo, elements in the world around us. Ergo, atoms. All right? Everything we can see, touch, feel, smell, perceive, it's all based off of the nature of an atom at its basis. This is mostly carbon. All right? The air you breathe has a lot of oxygen in it. Okay? Atoms make up matter. And then there's energy. Okay? Energy is the capacity to do work, to put matter into motion. And energy isn't really tangible beyond that. Like I can't have a handful of energy. That's not really the way that this works. Now, energy itself, taking up no space, there are different flavors, if you will, of energy. There is kinetic energy, which is involved in directly doing something. It's active. You're accomplishing a task. Oh, that's kinetic energy right there. And then there is potential energy, a storage. Potential energy is stored energy. You can store energy in a lot of different ways. Like our fella climbing to the top of this contraption here that is storing some potential energy that could then be released as kinetic energy, or alternatively, the batteries in my little clicker here, the batteries in this are storing energy that I can use to do things. That storage of energy would be potential energy. All right, potential energy. Now, there's a few more things to talk about here. There is chemical energy, like this battery. Certainly is chemical energy. That would be energy locked up in chemical bonds. We get this from the foods that we eat. And something like sugar would contain large amounts of chemical energy that we then will use to make a substance called ATP, adenosine triphosphate. That is our energetic molecule, okay? That is chemical energy as well. So when I flex my muscles, I'm burning ATP. That's chemical energy. There's also electrical energy. That would be energy flowing as charged particles. So the wiring in this room most certainly would be electrical energy. But your nervous system is your nervous system would be electrical energy as well. It's a flow of charged particles at the end of the day, and that's how this works. There is mechanical energy directly involved in motion. I can move around the room. That is displaying mechanical energy. And then last but not least, there is radiant or electromagnetic energy. That would be energy that travels in waves. This would be light. Okay? What you may not realize is we release heat. And the heat that we release from our bodies is most certainly radiant energy. Right? So energy versus matter, it is a worthy conversation. Now, speaking of the atoms that make us up, there are 6 elements in nature that comprise almost our entire body, 98.5% plus, I would say. These are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. We are composed of 65 plus 10, 75% oxygen and hydrogen. Let me say that again. 75% of your body is oxygen and hydrogen. How is that possible? What are you mostly made of? Oh, my southern dialect is giving this away ever so slightly. What are you mostly made of? You're mostly made of water. Okay? You are mostly H2O. That would be hydrogen and oxygen, the chemical basis of water. Your cells, you are mostly water. Ergo, lots of hydrogen, lots of oxygen. A lot of carbon in your system as well. 18% of your body by mass is carbon. That's because all of your large organic molecules are carbon-based. Anything that you consume or are made of, which is protein, which is carbohydrates, lipids, nucleic acids, they're all built of mostly carbon. And because of that, you contain a large amount of carbon. Nitrogen, calcium, phosphorus. Nitrogen is a limiting nutrient in a lot of things that we make. A lot of calcium and phosphorus in our bones. So these sort of things sort of make sense. But these are the major elements that make up who you are. And if I were you, I would have a good grasp of that. It's a very important concept to understand. All right, let's talk a little bit about subatomic particles. Now, these elements, made up of individual atoms, they're kind of smallest divisible unit, okay, an atom, the smallest unit of an element that maintains its basic functionality. These atoms are composed of smaller parts called the subatomic particles. These are protons, neutrons, and electrons. Now, I'm not going to talk a great deal about this. I will tell you that you can take a chemistry class and you can learn all about the periodic table of elements and sort of how all these things fit together. You can see entries on the periodic table and learn all about the numbers here. These tell you all about these protons, neutrons, and electrons. I simply want to point out the following, the basics of this. You see, an atom will look something like this. This is referred to as a Bohr model of an atom. In the inner portion of this, you find protons and neutrons. The protons and neutrons are found here because they are heavy and weighty, and they have mass, and they're held down deep in the core of the atom. Whereas the electrons are basically weightless. These electrons are flying around the outside of this thing like it's reckless abandon. So protons and neutrons deep in the core, they have mass, they're heavy, they stick together down in the very base of the atom. Whereas the electrons are just flying around in clouds all around the outer surface. This is important because when two atoms come together to bond to one another, what part of the atom is likely to interact? What part is likely to interact? The electrons. Chemical bonding is a relationship between the electrons of two or more atoms. Let me say that one more time. Chemical bonding, leading to molecules and macromolecules, is a relationship amongst atoms using their electrons on their outer surface. Much like if you and I interacted, if I shook your hand, our skin would be touching. In atoms, when they bond to one another, it's their electrons that interact and lead to that bonding. So this is a very important point of how all this fits. Atoms, you can see that there's a lot of different atoms out there. Okay, hydrogen, helium, lithium, beryllium, you name it. A lot of different atoms. You can always tell the difference. They have different numbers here at the top. This is their atomic number. You might as well keep this in mind. So an atomic number is this number here at the top of the periodic table. The atomic number tells you the number of protons. Let me say it again. The atomic number tells you the number of protons. which inadvertently tells you the number of electrons, because the number of protons and the number of electrons is pretty much always the same, okay? So here we got 20 protons, because the atomic number is 20, that tells us there's probably 20 electrons as well. So this is just a little breakdown of how the periodic table is structured, what atoms make us up for the most part, like carbon over here, all right, with its atomic number of 6. Carbon with its atomic number of 6, meaning it's got 6 protons, inadvertently telling us it has 6 electrons as well. They have balance in nature. This helps us to understand how atoms are structured and where we are going next, which is into chemical bonding, a relationship amongst electrons. All right. Now, there are a lot of terms that we could get into here, but I'm just going to jump straight down into chemical bonding. In the world of chemical bonding, we're going to talk about ionic bonds, covalent bonds, and then a side effect of covalent bonds referred to as hydrogen bonding. Okay? So ionic bonds and covalent bonds, these are different. And then a side effect of some covalent bonding called hydrogen bonding, which is very, 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 very important for us. Let's go here. All right, ionic bonds. We're going to keep this very simple. An ionic bond is where 2 atoms or more will come together and one of them steals an electron away from the other. Remember, all chemical bonding is electron-based. In an ionic bond, an electron is stolen away like a stranger in the night, okay? Taken from one atom and given to another. The perfect example of this is the relationship between sodium and chlorine. Sodium has a spare electron just hanging out in space, doing its thing way out on the outside of this atom. Chlorine has a whole bunch of electrons, but it needs one more electron to be balanced in nature. Okay? It really wants one more electron. Sodium's got one it doesn't really care for. Chlorine really needs another one. When these 2 kids come together, sodium donates an electron to chlorine, and chlorine thieves it away to never return it again. Now, what does this result in? Well, electrons bear a charge. Let me say that one more time. Electrons bear a charge. What's the charge on an electron? Well, electrons are negatively charged. You need to know that. Electrons display a negative charge. So if these 2 atoms are balanced, and then chlorine gains an extra electron, what's the charge on an electron? They're negatively charged. Chlorine gets a slight negative charge. Now, sodium used to be balanced, but now it lost an electron. It lost a negative charge. So sodium becomes overall a little bit positive. Positively charged sodium, negatively charged chlorine. It's like Grandma used to tell you. Opposites attract. These 2 will stick to one another. And as a result of this, we end up with this compound, sodium chloride, okay, sticking together, that no longer represents either of the 2 initial atoms. You see, sodium is incredibly dangerous. I don't have anything on here about it, but sodium is a metal, and if you place it in water, it literally will burn. You don't want to hold on to sodium. It would be very damaging and caustic to your skin surface. Super dangerous. Chlorine is in nature a gas. It's super, super terrifying. Chlorine was used as a chemical warfare agent. It was used to kill people in the past. Very dangerous is chlorine. You will put this in a swimming pool to kill everything that lives in that pool. Bacteria, viruses, what have you. Chlorine is dangerous. You bring these 2 kids together and you make sodium chloride. And sodium chloride is delicious. Sodium chloride is table salt. So the resulting compound is quite different from the initial atoms as a result of an ionic bond where an electron was stolen away to chlorine, resulting in a positively charged sodium and a negatively charged chlorine in their interaction. They stick together like magnets because one's positively charged and the other is negatively charged. This is an ionic bond. Next we have covalency. Covalent. Instead of an electron being stolen away, like in an ionic bond, in covalency, the electrons are shared. The electrons are shared. They're shared between two or more atoms, resulting in everyone there getting what they need in terms of the number of electrons required for that atom to be satisfied. It's a long story, but we're not going to go too deeply into the chemistry here. In this case, there is nonpolar covalency, and here I'm going to talk about polar covalency. Nonpolar covalent and polar covalent. These are two separate things that we've got to talk about. Nonpolar covalency. This is where the electrons are shared equally. Nonpolar covalency, equal sharing of electrons. What will result here is a very symmetrical molecule that displays no charges on its external surface. No charge. It's very neutral electrically. All right? Nonpolar covalency, equal sharing of negatively charged electrons. Because they are shared equally, no part of the molecule will display a charge. It displays as a nice, symmetrical, happy molecule. Then there are polar covalencies. In polar covalent bonding, well, one atom just likes electrons more than the other. Or one area of the molecule likes electrons more than another area of the molecule. Water is one of these. Water is H2O, okay? The oxygen side of this will love electrons, whereas the hydrogen side simply doesn't have the strength to hold on to the electrons as much. So the electrons hang around the oxygen side a lot, very seldom spending time with the hydrogen. And what's the charge on an electron? They are negatively charged. So if the electrons are hanging out over here all the time, this area of the compound displays a mild negative charge. If this is going to display mild negativity, then you must have a mild positive charge displayed on the opposing side. All right? So this is polar covalency. The rub to a polar covalently bonded molecule is that the sharing is not equal. There will be some areas of the molecule that display negativity, other areas of the molecule that display positivity. Now, let's jump back over here to ionic bonding. Here, an electron was stolen away. And the result of this is one atom that has a positive charge, one atom that has a negative charge, and they stick together like magnets. Here, in polar covalency, you have a whole compound displaying mild negativity on one border and mild positivity on the other. What happens if I bring together two water molecules? Well, if I bring together two or more water molecules, these water molecules begin to attract one another. They begin to attract each other, and you know this because if you dip your hand in water and hold it up, you'll see a bead form at the end of your finger. It's not individual atoms. No, that's incorrect. It's not individual compounds, molecules falling off your finger. Those molecules bind to one another. They stick together. Water likes to stick together. You may have seen a water strider, this bug at some stage in your life, stand on the surface of water. The tension generated by these compounds, by these molecules, pulling themselves together via these weak attractions from a polar covalently bonded molecule, pulling themselves together, resulting in tension that critter can stand on. We call these weak attractions between water molecules, we call them hydrogen bonds. We call them hydrogen bonds. Have you ever seen a car that's got a fresh wax job? All right. Water gets on the hood and it beads up into a little ball. That's because the water molecules are attracting one another and pulling together against gravity. Those weak attractions between water molecules are called hydrogen bonds. And this is really important because hydrogen bonding causes water to have some unique attributes that we're going to be talking about very soon. I've got a few here: chemical reactivity, thermal stability, adhesion, cohesion, solvency. We're going to talk about that in just a second. Let me just hit this real quick. Cohesion and adhesion. Cohesion means that water molecules like to stick to each other, and adhesion means that water molecules like to stick to other things. Like if I were to take this and dip it in water, water would stick to it because water likes to be cohesive. And the water molecules there would stick to each other because water molecules are adhesive. Or the opposite of that. Adhesion sticking to other things, cohesion sticking to each other. Water, because of cohesion and adhesion, because of hydrogen bonding, water has some unique attributes. And what are you mostly made of again? Because you are mostly water. That means that your body has the same unique attributes as does water in a lot of cases. Hydrogen bonding in between water molecules, in no small part, leads you to have some interesting attributes, basically allowing for life to survive as we know it. And we're about to talk about those interesting attributes in mere moments. All right, let's see. So just a little point to be made here. Grandma might have also told you at some stage that oil and water don't mix, and Grandma's not telling you a lie. There's a reason that oil and water are incapable of mixing. That's because water is polar covalently bonded, whereas oils are non-polar covalently bonded. They are incapable of mixing with one another because at a base level they are different in terms of their chemical bonding mechanism, if you will. All right, one's polar covalent, one's non-polar covalent. So let's make a little point there. All right, now we're changing gears. We're going to move into biochemistry now. Biochemistry is, let's see, do I have a book definition for you? Studying the chemical composition of and reactions associated with living matter. The way I broach this situation is simply to talk about inorganic molecules versus organic molecules and what makes them special. So we're going to begin that process now. First things first. An inorganic molecule is a molecule that does not contain carbon normally. There's virtually no carbon in an inorganic molecule. There are some weird exceptions, like carbon dioxide. Most certainly contains carbon. There are some acids and bases that have a little carbon in them. But the trick to an inorganic molecule is they don't contain much carbon, if any at all. And inorganic molecules are very small indeed. Compare that to an organic molecule. Organic molecules are carbon-based. They're basically just huge chains of carbon. Let me say that again. Organic molecules are huge chains of carbon. And organic molecules, as I just said, are massive. They're really, really large indeed. Organic molecules are incredibly large in size. Orders of magnitude larger than inorganic molecules. So we have these organic molecules, which by comparison are quite large. Okay, quite large. And some examples of inorganic molecules are salts and acids and bases, but water being far more important amongst these. And then the organic molecules being proteins, carbohydrates, lipids, and nucleic acids. Okay, nucleic acids. All right, as I was just saying, water is the most important of the inorganic molecules. Water makes up a huge percentage of your body by volume. Water molecules, which hydrogen bond to one another as a result of their polar covalencies. Polar covalent bonding and unequal sharing of electrons, resulting in positive charges in some areas and negative charges on other areas of these water molecules. If I'm not making myself clear, you need to know about this, team. Causing hydrogen bonds, causing water molecules that like to stick to one another, like on the hood of that freshly waxed car or in your cells, giving water some unique characteristics, like a high heat capacity, Water has a high heat capacity. That means it absorbs and releases lots of heat before changing temperature itself. That means that water is thermally stable. It takes a lot of energy to change the temperature of water. And that means that you are thermally stable. It takes a lot of energy, loss or gain, to augment your physical temperature. Why is that good? It's a lot easier to maintain your homeostatic temperature balance. You can walk outside in the really cold weather and check your mail, take your trash out, and not die of hypothermia, because you'd have to be out there a long time to really augment your body temperature. You can go out in blazing hot sun, and your body temperature isn't going to go up by much at all, because you are thermally stable, because you're mostly made of water, because water hydrogen bonds, and water is thermally stable. It's got a high heat capacity. All right, part 2 of this. Water has a high heat of vaporization. What that means is it takes a massive amount of energy to change water from a liquid to a gas. And you know this. You've probably tried to boil water before for cooking, and it takes an eternity, okay? What's the rub to this? When you get very hot, how do you deal with your temperature shifts? You sweat. You have evaporative cooling. When you sweat, the goal is for that water to boil off of your body. And when that water boils away, it liberates huge quantities of energy from your system. And you know this, because you've been swimming in the heat of summer, in July in Alabama, which is basically the surface of the sun hot. And when you get out of the pool and you're covered in water, you feel cold. Even though it's blazing heat outside, you feel cold because that water's boiling off of your body, liberating heat. Water has a high heat of vaporization, meaning that it takes a lot of energy to convert water from a liquid to a gas, meaning that your evaporative cooling mechanism is very, very efficient. Water is highly reactive. Water will participate in what are called hydrolysis reactions and dehydration synthesis reactions. Now I'm going to explain this in more detail here in just a second, but let's talk a little bit about dehydration synthesis and hydrolysis, hydrolysis. Hydrolysis and dehydration synthesis. Dehydrate. That means to take water away. Dehydration synthesis. Synthesis means to make something. Okay? So if it's dehydration synthesis, you make something by taking water away. So what's the opposite of that? Hydrolysis. If I tell you you need to go hydrate, I'm telling you need to go drink some water. Hydrolysis. Lysis means to break something down. Hydrolysis means to destroy by adding water to something. Okay, let's do that one more time. Hydrolysis, to break something down by adding water to it. Dehydration synthesis, to build something by taking water away. This is the foundation of your bodily function. When you eat foods, you break those foods down in your intestinal tract via hydrolysis reactions. All of your enzymes are hydrolysis reaction based. You add water to things and destroy them. How do you build your muscles? How did you grow taller? How did you build up this body that you have now compared to the little bitty critter you were back in the day? You did this through dehydration synthesis reactions. You take water away and build things. At a base level, this is how your body functions. Dehydration synthesis and hydrolysis, all of which function as a result of water and its capacity for hydrogen bonding and the fact that you're mostly water. Okay? Last but not least, water is a polar solvent. That's true. Anything polar, water tends to break up. That's not going to be as important for us here in human bio. Let's just jump right over into here. So water is an inorganic compound. You know this because it's very small and does not contain carbon. And pH scale, acids and bases. Acids and bases are also inorganic. They tend not to contain carbon and they tend to be very small. So I have a few examples. Bases like hydroxides as an example. Acids like your stomach acid, which is hydrochloric acid, HCl, no carbon there, are acids. So our bodies most certainly use things which are acidic and things which are basic. Acidic things are considered proton donors. When you place them in water, they release hydrogen. Okay? More hydrogen, the more acidic something is. So if you go and you buy one of those Warheads candies that's very sour, you pop it in your mouth and it tastes sour, what you're tasting is all that hydrogen ion being released in essence. Okay, it's very acidic. If you were to put vinegar on a salad, vinegar is acetic acid. Okay, these are acidic compounds. They have lots of hydrogen internally. Bases are proton acceptors. What that means is when you place them in water, they release what are called hydroxide ions. Bases are the opposite of acids. Okay, if you've had heartburn, that's a result of stomach acid kind of interfering with your esophageal lining, the lining of your esophagus above your stomach. Okay, stomach acid's not supposed to be there. So you might take some antacids to counter the acidity of the stomach acid. Antacid pills are bases. Your blood is ever so slightly basic. Your stomach is quite acidic. Okay? The urine that you produce is acidic. We also make other products that are basic, all right? So they exist in the world around us. Now, we measure acidity and basicity off of what's called a pH scale, and this is the pH scale. It runs from 0 to 14. If I were you, I would know this, okay? Things towards the 0 end are acidic. 7 is neutral. 14 is mega basic. So from 7 down, these are all basic. They would release hydroxide ions in solution. From 7 towards 0, these would be quite acidic, becoming more and more acidic as they go, with like lemon juice being around it too. An important point to this is that this scale is logarithmic. It's a log scale. So the difference between 6 and 5 is extreme. It's on a factor of 10. Okay, a factor of 10. So every number from one to the next is 10 times different than the previous one. So a pH of 5 is 10 times more acidic than a pH of 6. This is a logarithmic scale. These numbers are very different from each other. Okay, very different. Let's go here. All right, let's talk about the major organic molecules and see where this sort of takes us. All right, everybody, let's finish up here with organic molecules and get this thing taken care of. Our organic molecules are proteins, carbohydrates, lipids, and the nucleic acids. And the rub to an organic molecule is they are incredibly large and always contain carbon. Okay, always contain carbon. Before we really get into the conversation, a couple terms to get out of the way. Polymer and monomer. You need to be familiar with the nature of polymers and monomers. A polymer is a huge unit made up of smaller parts called monomers. This building is a polymer. The individual blocks in the walls would be monomers. Okay, mono means one, single. Poly means many, okay, plural if you will. Starch then is a blank of about 3,000 thousands of glucose blanks. Starch is a polymer of thousands of glucose monomers. Okay? Starch is built from glucose. Glucose is the building block for starch. Monomers and polymers. Here's two more terms you should be familiar with. Dehydration synthesis and hydrolysis. Dehydration synthesis. Dehydration synthesis. Synthesis, to make something by taking water away. Dehydration synthesis. You take the water away, you make something. Two monomers come together, you pull the water out, and you make a polymer. And then there is hydrolysis, to destroy using water. You take a polymer, add water in, and break it up into its monomers. When you grew taller, you were taking monomers, putting them together to build larger polymers. That is dehydration synthesis. When you eat a cheeseburger, you have to break down that cheeseburger through your digestive process into smaller parts and pieces. That is hydrolysis, taking something big and breaking it down into smaller subunits. This is the basis of your digestive reaction, okay? With that in mind, let's talk about some examples, shall we? First things first, carbohydrates. The carbohydrates. The idea of a carbohydrate is that these are sugars, okay? Carbohydrates are the sugars. And amongst these carbohydrates, there are simpler variants like the monosaccharides. Now look at this. Carbon, carbon, carbon. This is a big carbon ring. Big carbon rings. That's what this is. Lots of carbon and some hydrogen and some oxygen, etc., but mostly carbon. Big, huge molecules with lots of carbon in them. They're organic, okay? Here are the basic monosaccharides. Glucose, fructose, galactose being very important amongst these, the three major ones. And then there's deoxyribose and ribose that we'll be talking about when we do DNA and RNA. DNA is deoxyribonucleic acid. It's got a deoxyribose base. And then there's just ribose. Ribose is found in RNA, okay, which is a different thing altogether. Glucose, fructose, and galactose, you certainly get all these in your diet. Glucose is very important. I'd be putting a star by glucose here, folks. Glucose is incredibly important because glucose is your blood sugar, all right? Glucose is your blood sugar. When you eat a bunch of donuts, that's going to make your blood glucose levels, your blood sugar rise. So you release insulin to bring that back down. Negative feedback, okay? That sugar is glucose that's in your bloodstream. Someone whom is diabetic struggles to regulate blood glucose. If your blood glucose drops, you might have like a material to consume that is a glucose paste in essence to bring your blood sugar back up. Glucose is your blood sugar. Now, we can bring together some of these and form what are called disaccharides, monosaccharides, single sugars, mono means one. We can bring together these and form what are called disaccharides. Disaccharide means two sugars, okay? Like glucose plus fructose brought together forms what we call sucrose. Now, part one of this. You are familiar with sucrose. Sucrose is good old-fashioned white table sugar that you put in your coffee, all right? That is sucrose. Sucrose is made from taking glucose and binding it to fructose to form this disaccharide. That would be formed through dehydration synthesis. When you eat sucrose, you can't use it. It's too big. You can't get it into your cells. It's too large. So what you will have to do is use your digestive process to break sucrose down via hydrolysis reactions so that you can then absorb the fructose and the glucose independently of one another. Dehydration synthesis is how this is built. Hydrolysis is how your digestive process breaks it down. Right? Important point. Let's see, there's some other ones here. I've got maltose down here. Maltose is just two glucose molecules bound to one another. If you've ever heard of malted barley or malt liquor or any alcohol brewing, normally that's maltose. And then there's lactose down here, which you are probably familiar with as being in milk products. That would be galactose and glucose bound to one another. That is lactose. Again, lactose is too large for your body to utilize, so you have to break it down into its individual monosaccharides in order to absorb it using your digestive process, broken down through hydrolysis reactions. Let's go here. All right, you also will find that in the world around us there are polysaccharides. Polysaccharides are big old honking chains of monosaccharides all bound to one another. Massive units of monosaccharides, thousands of glucose molecules in a single starch molecule, for lack of better terminology. Cellulose, which makes up what we call plant fiber, is a polysaccharide. Peptidoglycan found in bacteria is a polysaccharide, but you don't have any of those. You, as a mammal, can only make one polysaccharide. And I would be very familiar with this if I were you. You are capable of making glycogen in your liver, primarily. When you consume huge amounts of sugar, you're going to start turning that sugar into glycogen and storing it away in the liver. Your liver will take up excess blood sugar and it will store it as a glycogen molecule because this glycogen molecule is so big it can't move around. It can't re-enter the bloodstream unless you tell it to. So when you have high blood sugar, like eating donuts, you will release insulin, and insulin basically tells the liver to take up excess blood sugar and store it as glycogen. And that's how this works. This is a storage form. Sugars are wonderful energetic molecules. So the sugar in your blood is there for a reason. It's there to help you be able to make ATP, your chemical energy. So the sugar is like the fuel that runs your car. And in essence, what that means is that glucose is the energy we use to build ATP that powers our muscles and everything else for that matter. Okay, you can hear me right now because you have ATP. It's a long story. You will store extra sugar as glycogen in your liver. It's just like putting money in the bank. Works like a charm. Let's go here. Let's talk about lipids. Lipids are fats. Oftentimes we think of fat, like in our bodily fat, as a long-term energy storage. So if you bring in huge amounts of sugar, you can actually recombine that and build it into adipose, into fats that you can store, everywhere all around your body. But generally speaking, lipids are fats. Fats and oils, phospholipids, steroids, all of these fall into the realm of the lipid, functioning for long-term energy storage. Now, the classic fat is a triglyceride. Triglyceride. Glyceride, glycerol. So a triglyceride is a glycerol molecule with 1, 2, 3 fatty acid chains. That's what these are called. And when they bind to one another by kicking off water, they form a triglyceride. This is dehydration synthesis. Triglycerides are the classic fat and oil. All right? And you may have heard the terms saturated fat versus unsaturated fat. Saturated fats are carbon chains in these fatty acid molecules that are completely saturated with hydrogen. It makes them very straight, very symmetrical, to be completely covered in hydrogen like this. Whereas there are also unsaturated fats. Unsaturated fats have weird bonding things that happen, so they are not completely full of hydrogen like here. It's all hydrogen to carbon. Here there's carbon-carbon bonding happening. And what that does is it causes the tail to have a kink to it. Okay? Saturated fats have straight tails. Unsaturated fats have kinked tails. Now let's talk a little bit about saturated fats and unsaturated fats. Saturated, these tend to be of animal origin, and they tend to be solids at room temperature. It's kind of like playing Tetris. If you've never played Tetris, you should. The straight pieces in Tetris, when you're stacking them up, are really easy to work with. They just stack up naturally. Whereas the kind of twisted pieces, they're hard to stack up by comparison. So saturated fats of animal origin, things like butter, they tend to be solids at room temperature because they stack up very easily to form solids because they're all quite straight. They look like this, like boxes. Whereas unsaturated fats tend to be of plant origin, and they have kinked tails, which means they don't stack up very easily. They tend to be liquid at room temperature. So plant origin, liquid at room temperature. Think about olive oil, a liquid at room temperature. Canola oil, corn oil. These are all going to be liquids. They are all of plant origin. They are unsaturated. Now I'm going to point out one more little grouping of things here. I'm going to point out omega-3 fatty acids and trans fats. Omega-3 fatty acids, which we tend to get from fish, or you can take like krill oil or fish oil tablets. You can even get this in milk and bread. Omega-3 fatty acids are considered heart healthy. They are good for you. They decrease the general fat load in your bloodstream, which sort of decreases your risk of heart disease. So that's very good. And you may have heard of trans fats. Trans fats are in essence incredibly dangerous for you. Trans fats are artificially straightened. They used to be kinked tails like this, and we will artificially straighten them to become a trans fat in a laboratory setting. And the essence of this is done in a lab. This is not a natural process, so you don't have enzymes that will allow you to break down trans fats. Now, it's good for the food industry because foods cooked in trans fats basically are immortal. Nothing can break them down. Ergo, they have a really long shelf life. But if you consume lots of trans fats, you can't break it down. So it just stays in your bloodstream and dramatically increases your risk of heart disease. So my advice, be light on the trans fats, okay? Be light on the trans fats. Not an ideal situation to consume large amounts of trans fats. In fact, the FDA made a statement about this and said that trans fats are not generally recognized as safe, which if you keep up with your government doublespeak is about as strong a statement as they will make. It is about as strong a statement as they will make on anything food related. So my advice to you is decrease your trans fat intake. Make sure you know what you're looking at in terms of food. All right, good. These are our fats. Let's go here. Let's talk a little bit more about some of these lipids. I want to talk briefly about phospholipids. A phospholipid looks like a triglyceride. Glycerol molecule, one fatty acid tail. Look at this one, it's straight, like a saturated fat. Another fatty acid tail, kinked, like an unsaturated fat. But then instead of having 1, 2, 3 for a triglyceride, this is a phospholipid. It doesn't have a third a third fatty acid chain. On the opposing side of the molecule, it has a phosphate group. It's a phospholipid. It's a lipid with a phosphate. And this makes it a very weird lipid indeed. Because whereas, again, grandma likes to say oil and water don't mix because oils are non-polar covalent and water is polar covalent, this oil has an area that acts like a fat, ergo it is non-polar, and then the phosphate end displays mild polarity. So this is a single molecule that is both polar and non-polar simultaneously. In other words, this area of the molecule can bind to water. This area of the molecule can bind to fats and oils. And that gives it some unique characteristics. Let's say I go out and I work in my shop and get my hands really greasy. Can I go inside and just run water and wash my hands and get the grease off? No, I have to use a little bit of soap. Soap goes on and the grease just washes right away. Because soap molecules are basically phospholipids. The phosphate group binds to the water. The fatty acid tails bind to the fats. And it allows us to take fats from our surface and wash them away as water. I invite you to cook some bacon and then clean the pan after. Put some hot water in that pan, put a little Dawn dish soap and start stirring. The water will go from clear to kind of opaque to almost milky white because you dissolve the fat into the water and it gives it that milky white appearance. Phospholipids are capable of this. That's not what we use phospholipids for. We use them for a very important purpose indeed. Phospholipids form up in what we call a phospholipid bilayer that makes up your cellular membranes. Your cell membranes are a double layer of phospholipids. The heads, which are polar and bind to water, and the tails, which are non-polar and work as a barrier. This is what allows your cells to be selectively permeable. Not just anything can get through a hydrophilic layer on the outside. Hydrophilic means water-loving, and a hydrophobic layer in the core of this thing. Hydrophobic means water-fearing. If I'm a polar molecule, I can get through the heads, but I can't get through the tails. If I'm a non-polar molecule, I can't get through the heads. Your cell membranes are selectively permeable. They only let in what they want to let in. They're a fabulous barrier because they're made of phospholipids. Interesting molecules. Let's go here. Let's talk about steroids. Now, all of these are going to be based off of cholesterol as a building block. Here is cholesterol, and here are a couple of steroid hormones. So cholesterol, steroid hormones. Cholesterol is actually very important for us. If I polled the class and said, hey, is cholesterol good for you or bad for you? Most of you would probably say bad. But the reality is, cholesterol is very important for us. I'm sorry, yeah, that's correct. Cholesterol is very important for us. We use cholesterol every day in our cell membranes to help them maintain fluidity. So it kind of keeps the membrane moving around the way it's supposed to move. And it is the building block. Cholesterol is the building block for steroids. All of our steroid hormones, like vitamin D, which allows us to absorb calcium from our intestines, like testosterone and estrogen, that give rise to our male and female secondary sexual characteristics. These are all based off of the steroids. You probably have heard of someone taking steroids to get stronger. What they're really doing is just taking testosterone. So if somebody wants to build muscle very quickly, they're in essence just going to take huge doses of testosterone, and that will increase their muscular build quite quickly. All right? This is not a good idea. Let me say it one more time. Don't do this. Real bad for the body. Real bad to artificially increase testosterone levels like that. Trust me. Let's go here. And then there are, of course, proteins. Oh man, proteins are so important. So important. Everything I see when I see another person is protein. Their hair, their nails, their eyes, their muscles. It's all protein. All protein. You have very important protein functions in your body. All enzymes that help you do chemical reactions, the contractile properties of your muscles are all protein-based. The oxygen carrier in your blood, this is very important. So in your blood, you have a molecule called hemoglobin in your red blood cells, in essence. And hemoglobin is a protein that carries oxygen. Okay, oxygen, hemoglobin, very important proteins, as it were. Now, proteins are made up of smaller subunits called the amino acids, and there's not that many of them, only 20-ish or so here. And it's kind of like the alphabet. There's not a lot of words in the alphabet, but you put them in different orders and make a lot of different words. Well, there's not a lot of different amino acids, but you can put them in different orders and make different proteins. Okay. Now, in terms of making proteins, proteins sort of follow this track in terms of how they are built and organized. All proteins start out as what's called a primary polypeptide. That would just be a big string of amino acids. No big deal. It's a string of amino acids. We're making protein here, team. First, it starts out as a primary structure. Next, it'll get molded and folded into a secondary structure. It can form what's called a beta-pleated sheet, which kind of does this sort of thing, or an alpha helix, or spirals. But what I want to point out about this isn't necessarily the structure. It's how this happens. Because all of these amino acid bonds here, these are all covalent. They are polar covalent, resulting in the option of having hydrogen bonding between the poles of these polar covalently bonded molecules. Secondary structures like these are held together with hydrogen bonds. All your proteins are held together through hydrogen bonding. All right? Your skin is held together via hydrogen bonding. Don't believe me? Heat your skin up and watch it fall to pieces when these hydrogen bonds are destroyed. This is called denaturing protein. If you go and you eat a salad with straight up vinegar, it's going to damage your tongue and you're going to feel like a dead layer on the outer surface. Again, if you burn yourself, it'll damage your cells on the outer surface. You are denaturing those proteins. You are busting up the hydrogen bonds that hold them together. Busting up the hydrogen bonds that hold these secondary structures as they are supposed to be, causing that protein to no longer be functional. All right. Primary polypeptide, string of amino acids. Secondary structures, held together with hydrogen bonds, beta-pleated sheets, and alpha helices. These can be further twisted into tertiary structures like this one here, where one of these will mold and fold upon itself. You can bring together several tertiary structures and form a quaternary structure. This is what hemoglobin looks like. This is hemoglobin. 1, 2, 3, 4. Tertiary structures come together to form a quaternary structure. Again, hemoglobin being the oxygen-carrying protein of your body, found in your red blood cells, carrying oxygen, hemoglobin. These are, or let me rephrase, this is how proteins are built. Hydrogen bonds holding together their unique structures with covalencies, polar covalencies, holding together their base amino acids. Last but not least are the nucleic acids. The nucleic acids store genetic information for the body and are capable of carrying genetic information from place to place. Now, the building blocks of the nucleic acids are called the nucleotides, and these are adenine, guanine, cytosine, thymine, and uracil. You are responsible for these. Okay, you need to know adenine, guanine, cytosine, thymine, and uracil. These are the building blocks of DNA. And a general nucleotide kind of looks like this. So what we have here is two nucleotides paired to one another. A nucleotide-based sequence is a phosphate, a sugar, and a nitrogenous base. A phosphate, a sugar, and a nitrogenous base, hydrogen bonding to one another in the central core here. This is thymine bonding adenine. Thymine and adenine. Now, you probably have a grasp of this already, but DNA is in the form of a double helix. It looks something like this classic twisted ladder, as it were. The classic twisted ladder, the double helix of DNA. Now, what I want to do right this second is I want to have a little bit of a conversation about the differences between DNA and RNA. Okay? And I guess we could talk about who binds to who, but differences between DNA and RNA is my main goal. I invite you to read this. Feel free. I'm just going to explain it to you. So let's talk about the differences between DNA and RNA. All right, it's going to go something like this. DNA is like the cookbook for how to make proteins. That's all it's for. DNA is just code for how to make proteins. You have DNA in your cells, and people think of DNA as this mystical storage of all of who you are. It makes me who I know. No. All DNA is a cookbook for making proteins. There's nothing fancy to this, team. Nothing fancy. Cookbook for making proteins. Always found inside the nucleus of your cells. That's where you're going to find DNA. RNA is very small. DNA molecules are huge. RNA molecules are tiny, teeny tiny little things. And there'll be just a little tiny, teensy copy of a single spot in DNA. They can leave the nucleus and go out into the cell and say, hey, make this protein. Imagine we were running a kitchen and we had one massive cookbook. I'm going to make a cake. You are going to cook a steak. And yes, that rhymed. I don't know why. I'm going to come in, I'm going to take my phone out and make a copy of the recipe for that cake. Then I can leave and go out into the kitchen where the work gets done. That's RNA. RNA is a copy of a single piece of DNA, a tiny strand, very small. Whereas DNA molecules are massive, double helixes, double-stranded. RNA molecules are tiny, small, single-stranded, and they're capable of leaving the nucleus to go out and get the work done. All right? DNA, big. RNA, very small. Let's make sure we're satisfied here. A couple of things to talk about. Again, DNA is deoxyribose as a sugar, okay? Whereas RNA is just ribose. DNA contains adenine, guanine, cytosine, and thymine. RNA contains adenine, guanine, cytosine, and uracil. RNA contains uracil. DNA contains thymine. That's a good point to be made here. And for my purposes, I'm going to go ahead and make you know who binds to who. Let's just steal the camera away, shall we? All right, come here. The way I remember this goes something like this. Straight-sided letters like adenine always bind to straight-sided letters like thymine. All right? Curvy letters like cytosine always bind to curvy letters like guanine. Adenine binds to thymine, cytosine binds to guanine. And that's how this works in DNA. All right, this is in DNA. The only variation is from this uracil in RNA. In RNA, there is no thymine. No! That's going to be replaced with uracil. Okay? So uracil replaces thymine in RNA. And that's how this works. When we do cells, when we talk about genetics, we're going to talk more about this. For right now, I just want you to learn who binds to who. All right, team, that's going to be the end of our first lecture. I hope you thoroughly enjoyed yourself, and I look forward to seeing you in the next one. Thank you.